Hey, what's up, biology students? Welcome to the virtual lecture on cellular respiration and fermentation, part 2. In this lecture, we're going to cover the two final stages of cellular respiration, those being the electron transport chain and a process called chemiosmosis. First, I would like to do a brief recap of our previous video, which focused on stages 1 through 3 of cellular respiration, those being glycolysis, pyruvate oxidation, and the citric acid cycle. And to briefly summarize what occurred during the first 3 stages is to say that we took glucose, right, a small digested biomolecule, and through a series of chemical reactions, which are in reality quite complex, we use enzymes to break down glucose by a chemical reaction called oxidation. And in this process, we stripped or removed pairs of electrons from glucose as we broke it down. Not only that, but we harvested these pairs of electrons from glucose, and they were captured by a special molecule called NADH, which we call the electron shuttle. So the whole goal of stages 1 through 3 is to just completely break down glucose until there's nothing left, until there's no energy left, until you're just left with 6 molecules of carbon dioxide, and a bunch of electrons. So the net result of the first 3 stages is waste, a bunch of CO2, which you exhale into the atmosphere, but more importantly, several molecules of NADH, specifically 10 per glucose, each NADH molecule carrying 2 high-energy electrons from that original glucose molecule. Also, plus 2 molecules of FADH2, but for simplicity, let's just focus on NADH. So our question moving forward is, wasn't the whole goal of cellular respiration to produce ATP? You know, we've spent a lot of time talking about this process, and so far, I haven't heard anything about a tremendous amount of ATP being produced. So when's that going to happen? How's it going to happen? Where's it going to happen? That's the focus of this video. So we're going to be discussing this process. How do we turn all those molecules of NADH, specifically their electrons, into many molecules of ATP, which we can finally use to fuel activities that require energy, like, for instance, movement. You know, as a teacher, I always care about the big picture. To me, you know, I may ask you this, but I don't care if you know that there are 6 molecules of NADH produced per glucose during the citric acid cycle. That's kind of just like a meaningless number that you're probably going to forget right after you take the test. It's much more important to me that you understand, for instance, how this process of cellular respiration relates to your everyday life, how you might use it in a future biology class. Okay? Everything I teach is for a reason. I try to avoid busy work or meaningless topics. So that's why I keep referring to this picture. I want you to definitely understand how cellular respiration relates to eating, how it relates to nutrition, how it relates to digestion, where it fits into all that. Because if you don't understand that, then I feel like there's no point of talking about any of this. Okay? So that's the most important thing to me, is the big picture, how this fits into your metabolism. So, again, this is what we're going to be focusing on, the latter stages of cellular respiration. Now, I want to point out that we talked about earlier in the previous video that we can summarize cellular respiration as the transfer of electrons from glucose, a food molecule, to oxygen. Now, like we said, they don't go directly from glucose to oxygen. That would release all of their energy, and it would be like exploding that molecule of glucose, releasing all of its energy at once. We don't want to do that. we want to slowly release the energy of our glucose so we can store it in these nice little molecules of ATP. We don't want to explode our glucose. So like we said, those electrons don't go directly to oxygen. There's a few steps along the way. So far, we've only seen them take one step, from glucose to, sorry about that, from glucose to NADH. But we're not quite to oxygen yet, and still we haven't made any ATP. We made a little bit in glycolysis, but that's negligible. So the point of this drawing here at the bottom is to say that the electrons that are being held by NADH, they still have a lot of energy, okay? If we imagine that here's glucose, the electrons when they're located at glucose, here's the electrons once they get to oxygen, here's more energy, here's less, okay, we're going this way. We're only here. So we haven't dropped off very much, so those electrons still have a lot of energy. Okay? So we're going to fill in the gap here. What goes on from here to here? Okay, so let's go to our slides. We're going to start with stage 4, which is the electron transport chain. And we're going to start with all of those molecules of NADH and FADH2 that we produce during stages 1 through 3. And we're going to follow them to the inner membrane of the mitochondria. And that is where stage 4 is going to occur. Now, I'm going to use the term ETC oftentimes to represent electron transport chain, just so it's easier to say. So if I say ETC, that just means electron transport chain. And what we're going to see here is that all of these molecules of NADH and FADH2 are going to deliver their pairs of electrons to the ETC. So they're going to let them go, leave them behind. And once those shuttles have deposited their electrons, they'll go back. And, you know, you're constantly breaking down food, so you're not just breaking down one glucose. So you're breaking down, you know, hundreds, thousands at a time. So they're going to go back to glycolysis, go back to the citric acid cycle, and pick up another pair from a new molecule of glucose that's being broken down. So these shuttles, that's why they're called shuttles, they go back and forth between stages 1 through 3 and the ETC, carrying electrons. So that's what we're going to talk about here. First thing we want to do is go to our cell diagram, specifically focusing on the mitochondria. And I want to fill in some parts here. This is our cell membrane in black. Now again, we're just focusing on a specific part of the cell as it relates to the mitochondria. There's other stuff going on, obviously. The region within the cell is the cytoplasm. These little yellow circles with the minus sign inside, these represent electrons. So this is a molecule of NADH carrying a pair of electrons that it attained from glucose. What else? This whole organelle here is the mitochondria. Now again, this is somewhat of a review from our previous video so far. Let's label some more. In dark red, we have the inner membrane of the mitochondria. Remember, it has two membranes, one here depicted in orange, and a second one depicted in red. In between the two membranes, we have the intermembrane space. And then finally, we have the compartment that is inside the inner membrane, and that's called the matrix. So the point of this drawing is to show you where the electron transport chains are located within the mitochondria. So the electron transport chains, as we're going to learn, are a series of molecules and proteins that are really good at transferring electrons, passing them along, and allowing those electrons to become progressively more stable. So slowly releasing the energy of those electrons as they're passed. We're going to use that energy to make ATP. So each of these collections of little colored blobs, orange, pink, green, and blue, that represents an electron transport chain. And the first thing I want you to recognize is that the electron transport chains are embedded. They're located within the inner membrane of the mitochondria. I've drawn two of them to emphasize that you don't just have one electron transport chain per mitochondria. You have thousands of them embedded into the inner membrane. Obviously, I didn't want to draw thousands, so I drew three. But let's just label these as ETC. It's the same thing. And for now, that's all we're going to label here. We're going to come back to this drawing. So now we understand where stage 4 is happening. It's happening along the inner membrane of the mitochondria. Okay. So what happens at the ETC? Well, first, our shuttles are going to, like I said, release their pairs of electrons. Second, those electrons are going to get passed from complex to complex, or from protein complex to protein complex, down the ETC, until eventually those electrons get passed to oxygen. And as we're going to see, once oxygen gains a pair of electrons, it gets reduced into water. So the oxygen that you breathe in from the atmosphere ultimately gets converted into water once it attains 2 electrons from food, like glucose. Kind of an odd concept. Okay, so one important idea here is that as these electrons are passed along the ETC, energy will be released. So that is because each molecule allows these electrons to be in a slightly more stable location, oxygen being the most stable. That's important because we're going to use these little packets of energy represented by steps here. We're going to use that energy to build ATP, and that's the overall goal of this entire process. Now, I know this is hard to understand verbally. We're going to draw this all out in a moment. But I want to say one more thing. These small packets of energy that are released, it's not going to directly be used for making ATP. We're first going to convert that released energy into a steep electrochemical gradient across the inner membrane. And it will finally make sense why I spent so much time talking about electrochemical gradients in the membranes module. I did so for a reason, because it's a heavy component of making ATP. So if you remember that from the end of the membranes module, that will really be helpful here. Okay. Here's a textbook diagram of the ETC. I have my own diagram. It's going to be the same thing, just a little more colorful. So We're going to take the time to go through my diagram of the ETC. We're going to draw it out. And then most importantly, we're going to follow the path that a pair of electrons takes from NADH until finally it gets to oxygen. Now remember, it's analogous to electrons falling down a staircase or jumping off a diving board. So electrons here at NADH, this is kind of, uphill. Oxygen is downhill, more stable. Okay, so we kind of have a flow of electrons downhill. And it's really analogous to how a water wheel works or a hydroelectric dam works. And I have a picture for that if you're interested. It's basically allowing water to flow downstream using the force of gravity. And as that water flows downstream through the dam, it releases energy. It's like a rush of motion, and it spins something. In a water wheel, it spins something, for instance, to mash up grain or to make iron, kind of a primitive thing that was used pre-1900s. But in a dam, water flows and it spins a turbine, which moves a magnet pretty much around wire, which generates electricity. And it's strikingly similar to this process. So if you replace the flow of water with the flow of electrons, you're pretty much doing the same thing. So I want you to start to imagine these complexes of proteins, which I depicted as orange, blue, green, and red in my other diagram. Picture them as wheels or turbines, and picture the electrons as water flowing downstream. And as the electrons pass through these wheels, it's going to spin the wheel. And we're going to use that spinning motion to do something. So that's why I have the wheels there. It's an analogy to make a very difficult process hopefully more easily understandable. Okay? So let's go to the diagram and draw that out. So first off, I want to say, well, what are we looking at here? Well, if you imagine that we zoomed in along the inner membrane here in red, that's what we're looking at here. So this is that red membrane, okay? Just a more zoomed in view. So the first thing I want to do here is I want to label the major parts of this diagram, starting with the membrane itself. And then outside of the inner membrane is the intermembrane space up here, which I already have labeled. So that's this whole region, is the intermembrane space. Inside the inner membrane is the innermost compartment of the mitochondria, and that is called the matrix. And that is represented by this space. So that's where we're at. What else? We have a little key here on the side. The little gold circles, those represent electrons. And the little pink circles with the H+, those are protons. Also known as hydrogen ions. They are charged. They have a +1 charge. Okay, so let's label the ETC itself. The ETC is composed of really large complexes of proteins and enzymes, which we're not going to get into the details of it. We're going to draw them in an easier way, a more cartoon way, with my colored blobs. And there are 4 of them. The first is called protein complex 1, and they use Roman numerals for whatever reason. The one in red is protein complex 2. The one in green is 3. That was a little messy. That's a 3. And then finally in blue, number 4. one other thing I want to label, in case you don't already know at this point in the class, O2 stands for molecular oxygen. It's the oxygen that you're constantly breathing in from the atmosphere. So that is oxygen. And you have a bunch of oxygen inside your matrix. So when you breathe in oxygen, it diffuses from your lungs into your bloodstream. It is transported by a protein called hemoglobin and then dispersed to all of the cells in your body. It goes into their cell membrane, then it diffuses into the intermembrane space, and then finally it diffuses from high to low until it gets to the matrix. So you have this huge buildup of oxygen inside the matrix of all of your mitochondria. That's really important because it is a key component of the electron transport chain. Specifically, it is the final step in the electron transport chain. A few more things you're probably wondering what they are. We have these right here, Q and Site C. Okay? What does that stand for? The name's not that important. I don't want to give you a bunch of complex sounding terms, but they do have names. This is called ubiquinone. I'm just going to call it Q for short. This is called cytochrome C. We're going to abbreviate it. These are electron shuttles that are analogous to NADH. They're going to help us move electrons through the ETC, eventually arriving at oxygen. Okay? So think of them as shuttles moving left to right. So it's going to move, it's going to pick up an electron, move this way, move back. It's just going to shuttle left to right, left to right. All right, so that's what those are. So let's do this. Next thing we want to do is trace the flow of electrons down the ETC. Now, let's always keep in mind our analogy. So I know there's no hill here, but the left side of the ETC, this is in a sense at the top of the hill or the top of the diving board. Over here at oxygen on the right side, this is downhill or down the staircase or, in the water down from the diving board, okay? So if we move from uphill to downhill, we can release energy, right? That's what we're looking to do. So let's follow those electrons. So the one thing I can't do with this diagram because it will erase other key components is I can't erase the electrons as they've been transferred. I'll show you what I mean in a second, but let's start. So step number 1. NADH is going to transfer its pair of electrons to the ETC. They will first pass through complex 1. And then they will arrive at this shuttle called Q. So that is step number 1. So I'm going to draw the result of that. That's why I have these electrons up here. I want to use them. And let's imagine that one of those electrons did just that. Now what I'm saying is I would like to go back here and erase this electron, but I'm not going to do that the whole way through because it's too touchy. So just imagine it's not there anymore. Lastly, let's draw our arrow. That is the path that the electron took. Now, something really important happens here. Remember earlier I said to imagine these protein complexes as wheels or turbines that once this electron passes by downhill, it's going to release some energy and it's going to spin it. Okay? So think of this as a wheel that gets spun as that electron passes by. And the protein complex is going to use that energy that was released as the electron passed to do something really important for us. And that something is to pump ions across the inner membrane, from the matrix to the intermembrane space. What ion is going to get pumped? Protons, hydrogen ions. So I'm going to move that here. Okay. Now, I should start by saying this. Let me draw this out. There is a high concentration of protons in the intermembrane space relative to the matrix. So there are a lot more protons up here than down here. And what I just told you is that this protein complex just pushed and pumped a proton in this direction. Now think back to membranes. Does it want to go that way? Do things like to move from low to high? The answer is definitely not. Things in the universe like to spontaneously spread out from high to low. They don't want to go from low to high. So it requires energy to do that. Where did this complex get the energy to push that proton? Well, from the passing motion of that electron passing by. Or if you want to draw it this way, like that, downhill. Okay? So this is, in a sense, that wheel that just got spun. And we use that motion to push that proton across the membrane. Now we're basically going to do this same thing 2 more times. So if you understood that somewhat, we're just going to repeat it. So next, we're going to take this same electron and it's going to get passed from Q. In the process, it's going to pass through complex 3, spinning it, allowing it to pump a proton. And then finally, it's going to end up at this carrier, cytochrome C. A little bit further downhill, a little bit more stable. So let's draw that out. Now our electron is here. We, in the process, released a little bit more energy, a smaller packet of energy. We did this. That electron just passed by, right, really fast. It released some energy. And that energy is going to cause our wheel, if you will, to pump some more protons into the intermembrane space. So we're going to draw that. And then the same thing is going to happen one more time. That same electron is going to take one more transfer. It's going to go from cytochrome C. It's going to pass through complex 4, spinning it, and then finally it's going to reach its final destination, which we call the final electron acceptor, aka oxygen. And now you're finally going to get to understand why you're constantly breathing oxygen, why it's required. Well, it's a part of your ETC. And as we're going to see in our next video, we're going to see the effect of not having oxygen around. And as you're going to see, this whole process shuts down. So you need oxygen here to gather electrons in a sense. And let's finish our drawing. So it's not too bad. There's just 3 electron transfers. Each time they're transferred, they pass through a protein complex, allowing that complex to do something that requires energy, in this case, to pump protons against their concentration gradient, generating something called a steep electrochemical gradient, which is just a really fancy way of saying there are a lot of protons on this side, relative to this side. You're probably still wondering, why? Why are there so many steps to go from food to ATP? Why can't it be a little easier? Well, I wish it was too. I didn't make this stuff up. It just kind of is what it is. But we're almost there. There's one more step to getting to ATP. Because you're probably looking at this and you're like, isn't the whole goal of this process to make ATP? And still it hasn't happened yet. So what are we going to do with this gradient of protons? And how is that going to result in ATP? That's the final step. That's stage 5, chemiosmosis. Now, I want to finish drawing this gradient, so bear with me. I'm going to make some more protons here. I just want to show that there's a lot up here. High concentration. And what creates this high concentration? The ETC does. Protein complexes 1, 3, and 4. Relative to this side, where there's a lower concentration. So I want to ask you this, just looking at this picture so far. Where do the protons want to go? Do they want to go this way? Or do they want to go that way? Think back to diffusion. All things like to move from an area of high concentration to an area of low concentration. And there is a really high concentration up here, a very low concentration down here. So these protons, it's like water behind a dam. Okay, they really, really want to go to this side. And if we open up a little passageway for them to do so, they'll shoot right across that membrane with a tremendous amount of force. And we can use that force, that flow of protons, to finally make our ATP. And that's what we're going to do. But we're not quite done with this drawing. This is a very important drawing. It is really the only drawing of this video. Represents stages 4 and 5 eventually. But I want to show you this, that once oxygen gathers a pair of electrons, which I'll show you right here. Remember we said that it gets converted into a new molecule, becomes something different. So what is the name of, I don't know if I can erase this cleanly, what is the name of oxygen once it's gained 2 electrons? Good enough. Water. This is now water. So if you remember back to the overall equation for cellular respiration, you start with glucose, you start with oxygen, and you come out with CO2, ATP, and water. This is where the water comes from. Your oxygen gets turned into water. Now you may be wondering, why do I need to drink water if I can just eat food and that food causes me to create water? That sounds great. You do produce some water doing that, but it's kind of a negligible amount. Remember, most of your body is composed of water. You need a lot of water, and this is not that much. So you cannot survive by just doing cellular respiration. Not enough water. But that's a product. I wouldn't call it a waste product because we do use water. What else did I want to show you? One other thing you might be wondering looking at this picture is, why on earth is there a complex 2 if you're just going to skip it? What's the point of it? Now I won't test you on this because this is a intro class and I like to keep it as simple as possible. But I do want to point out, remember we do have these other shuttles, a few of them, 2 of them, called FADH2 that were produced during the citric acid cycle. So we can say that. Remember, we have 10 of these from stages 1 through 3, and we had 2 of these. So this is what we're going to use them for, this process. Complex 2 comes in here. I'm not going to draw the arrows because I don't want to make the picture messier, but I'll show you with my laser. FADH2 does not donate its electrons here. Instead, its electrons get passed to complex 2, which then go directly to Q. So it takes a slightly different path as such. And that path misses complex 1 and results in less protons getting pumped. So in a sense, these electrons result in slightly less ATP production. Not ultra important, but it's worth pointing out, because otherwise you're just left wondering what on earth was the point of any of this. That's why. But I'm only going to test you on NADH. Also, this complex 2 is not a pump. Only 1, 3, and 4. Okay, that's huge. That's a lot. This is all very important. So I think the next thing I want to do is I want to show you this in motion. I kind of showed it in motion, but I can only do the best I can. Let's watch an actual video of it. So let's see. Should be coming up soon. But this is basically the same thing that I drew. Hopefully you liked my diagram better than the book, but same thing. Not testable material, but just this is the analogy of the ETC and how it relates to a dam or a water wheel. Same kind of concept. You know, I don't have the video, or I'm not telling myself to show the video at this point in the lecture, but this is when I want to show it, so I'm going to go over here and pull it up while it's fresh in your head. Let's check this out. We're just going to watch the ETC. And we'll watch stage 5 too as an introduction.
In order to understand how the majority of the energy is produced by aerobic respiration, we need to follow the NADH and FADH2 molecules to the next stage, the electron transport chain. The electron transport chain is a...
Sorry about that. Wondering why it's blurry. It seems to be completely downloaded. I'm going to go with it because I have to.
DH and FADH2 molecules to the next stage, the electron transport chain. The electron transport chain is a series of membrane-bound carriers in the mitochondria that...
Sorry to interrupt. This in blue, I know I don't have my laser here, but the one on the left in blue, that's complex 1. The one in purple is complex 3, and the one in teal, second furthest to the right, is 4. They're just telling you the fancy names of these complexes. The one in green is Q. The one in orange is site C.
Pass electrons from one to another. As the electrons are transferred between the membrane proteins, the cell is able to capture energy and use it to produce ATP molecules. Proteins in the chain pump hydrogen ions across a membrane. When the hydrogen ions flow back across the membrane through an ATP synthase complex, ATP is synthesized by the enzyme ATP synthase. Oxygen acts as the terminal electron acceptor. By accepting electrons, oxygen is reduced to form water.
Okay, so a lot going on, you know, when it's in motion, but I want to point out that the electrons are kind of like those bright blue spheres. So right now they're on the far right side. The 2 little red dots, that's oxygen. And I want you to watch how that oxygen is going to become water right when I press play again. It's going to gather 2 electrons and become water. Also, the protons are the little purple spheres with the plus signs in the center.
Byproduct of the electron transport chain. All the high-energy electron carriers from the previous stages of cellular respiration bring their electrons into the chain. From this, the bulk of ATP from the entirety of cellular respiration is produced, a net of 32 to 36 ATP. In summary, we have seen how the 4 stages of cellular respiration are responsible for converting the energy found in the glucose molecule into ATP, the energy battery of the cell. On average, 36 ATP molecules are produced per glucose molecule that entered the cell. In the process of producing ATP, oxygen is brought in from the bloodstream to be the final electron acceptor in the electron transport chain, and the carbon dioxide that is produced as a byproduct is released. The goal of cellular respiration is to transfer the energy from the food that we eat daily into ATP that our bodies can use. This process starts with the eating of a snack or meal and ends with capturing the energy from the complete breakdown of the nutrients into energy and carbon dioxide.
All right, so the only thing we have left to cover here is that final process of using that special enzyme called ATP synthase to use the proton gradient to build ATP. So that's where we're headed to. This slide is just emphasizing the importance of oxygen. It is the final carrier in the ETC. It is the final destination of our electrons that originated from our food, like glucose. That is why we call it the final electron acceptor. This is just, it's a lot of writing to describe everything that we've drawn out and just watched in that video. So I'm not going to be redundant and waste your time. It's just a nice summary in paragraph form of everything that we've covered in the last 20 to 30 minutes. So nothing new here. I've already said all of this. Important though, to read it again for review. Here at the bottom I say, let's draw out this very important steep proton electrochemical gradient that was generated by the ETC. And I said, let's do it on 2 diagrams. Now the nice thing is we've already done it for one of them. That was this one. Let's do it also over here on this more zoomed out, if you will, depiction of the entire mitochondria. So again, I'm going to make some more protons. Because again, this is the same view of what we were just looking at, just more zoomed out. So maybe a little bit more difficult to squeeze these in here with my finger, but hopefully I can get it done. So far, so good. Again, what I'm trying to illustrate is that there is a really high concentration of protons in the intermembrane space relative to the matrix. What created that gradient for us? Protein complexes 1, 3, and 4 of the ETC, which used the gradual release of energy from falling electrons to do so. So that's enough to get the point across. Let's just show that there's a smaller, lesser amount here. And that's it. We can finish that off by just saying, squeeze it in here. High concentration in this space. Low here. And again, we said that's important because things like to naturally, spontaneously move from high to low. So all of these protons really want to get back inside of that matrix. Now the question is, how do we let them do it? Because when they do it, they're going to release a ton of energy. And that, in a sense, is the same energy from your food. And if you go back far enough, it's the same energy from the sun. So kind of crazy. So how do we use that gradient to make ATP? Well, the process of using that gradient to make ATP has a fancy name. It's called chemiosmosis, as you can see right here. I don't really like the name, as I don't with a lot of things in biology, because it tells you nothing, or at least from what I know, about what's going on. You would think osmosis, that there's a movement of water. It's not really happening. So it's a bad name, but you need to know what it means. This slide here is just summarizing the gradient of protons, which we just drew twice. So here's a third view of the same thing. All right, so as I alluded to, we need to open up a path for these protons to get back where they want to go, the matrix. And thinking back to membranes, what is a perfect little tunnel or channel? I just said it, oops. What's the perfect path for an ion to diffuse through the membrane? Because remember, ions are charged. They cannot pass through the membrane even though they want to. They're blocked. So we have to open up a protein, here's the answer, a channel, to allow them to pass across. So my question is, what is the name of this channel? Well, the channel, shown here in dark purple, is part of a large protein/enzyme/channel called ATP synthase. Probably the most important protein on the planet, because it manufactures our ATP, which powers all life on Earth. That's what this guy does right here. So how does it work? Well, basically it unleashes the energy of this gradient. So number one, it opens up a perfect little channel for these protons to pass through the membrane. But they don't just shoot right through the channel. They take kind of a roundabout way. They actually latch on to ATP synthase, and they take a little rotation. They spin with it, okay? So they go whew, like that, and then they get dumped into the matrix. So this flow of protons is converted into mechanical energy, spinning. And it's kind of cool. You can actually look at videos of ATP synthase that has been like tagged with a fluorescent molecule so you can see it glowing. And they attach something to the ATP synthase, like a, I don't know, like a rod or something, and you can watch the ATP synthase spinning in real time. Basically, it's a little machine. This is like the rotor of the machine, like a little, I don't know how else to say it. And, you know, what this machine does is it manufactures ATP. So that spinning spins the enzyme region of ATP synthase. A lot of complex chemistry going on that's even over my head, but basically it opens and closes active sites, and that force allows your ATPs to be put back together. Finally, we finished this process, right? Food to ATP, there we go. And it's really efficient. Per glucose, you get about around 28 to 32 ATPs produced via ATP synthase. That's a lot. All right, so another one of those slides where it's basically everything I just said in paragraph form. Let's just take a quick look at it and see if there's anything new here. And there is one thing that I want to emphasize, and it's this portion right here. And this goes back to the whole broader view of this entire process. Why are we doing this? How does it relate to food? How does it relate to ATP? How does it relate to functions like movement? Okay? And remember, the whole goal of this process of cellular respiration is to turn small digested food molecules like glucose, release their energy, so we could put our ATPs back together, so we can recharge them in a sense, so we can continue to do everything that we do as a living thing. So we're looking to use that energy released from glucose to do this. Take our spent ATP, aka AGP, and build it back together into a brand new charged ATP molecule that we can use again. I want you to realize that's exactly what this enzyme does. It puts your ATP back together. In a sense, it builds ATP. Do you also recognize that this is the reverse of ATP hydrolysis? So this is half of the ATP cycle, right? You can then go and use this ATP to, say, for instance, move. Then you'll be left with ADP. Well, fortunately, it's reusable. We can use the energy that we just talked about from food to put it right back together. And it's a constant cycle for your entire life. Constantly fueled by the food that you eat. If you stop eating food, you can't put these back together. So that's why you eat. What else? Nothing else here, at least. Except for, let's draw out this process of chemiosmosis on our diagram. So, what are the purple blobs? Those represent ATP synthase. I drew 3 of them. There are many more than 3 located on the inner membrane, but I don't want to draw more than 3. So that's what it is. Let's quickly draw what it does. If I can squeeze it in there. I think I'm going to choose this one right here because I have some room. First step of chemiosmosis. Very important. A proton, I know it's kind of drawn kind of big relative to the ATP synthase, but bear with me. That proton is going to pass through ATP synthase. Not only pass through, but it's going to spin it and cause it to rotate. That rotational force is used to do a chemical reaction that requires energy. In this case, plopping that phosphate group back onto ADP, regenerating ATP. And I want to put this in red because it's so important. It's the whole goal of this entire module was to do just that, produce ATP. And then finally, that, I can't draw it because it will get messy, but that proton is going to reach the matrix. That's it. So you have these protons that are constantly flowing through your ATP synthases, constantly causing them to spin and pump out ATP. Whew, right? Seriously. I don't like to say like some things are harder than others, but this is probably the toughest concept in biology. Cellular respiration. Hopefully, I made it easier to understand. So here I have some more videos. You can watch the entire video of the one that we just watched a few moments ago. Here's a second one that talks about both ATP and cellular respiration. Here is a textbook image of both the ETC and chemiosmosis all going on at the same time. This term here, which I never said, oxidative phosphorylation, you don't need to know it. I'm not going to have you responsible for it. It's just a fancy term for stages 4 and 5 collectively together. This is a really nice slide when you're studying for this module quiz. It summarizes the net products of all the stages that we've talked about. And these are some things you want to know for quiz questions, like the net products of glycolysis, pyruvate oxidation, the citric acid cycle, and then finally, stages 4 and 5. Everything here is very important. It's a nice summary of everything. But nothing new, so I don't want to waste any more time on it. This is our last slide for this video. And it's talking about the ATP cycle again. And again, we said ATP is a reusable source of chemical energy. And the reason that we eat food is to release the energy from those small digested food molecules. And we use that released energy to put our ATP back together. So if you want, in a simple sense, you could summarize everything that we've talked about in the last 3 videos as such. You eat food, you digest it, you release energy, you use that energy to put your ATP back together. That's it. Then, if you so please, you use that ATP to do things, communicate, move, build, pump, whatever you need to do. And it's a cycle. And since it's so important, I want to emphasize that it was here all along. I just didn't say it. The ATP cycle is depicted here. I'm trying to get my laser to work. Here it is. This. So I've taken you through this whole path, and I can get rid of these question marks since we just finished. Now it shouldn't be such a mystery with how we go from electrons carried by NADH to ATP. Now we finally have our $10 bills. We can use those $10 bills to do nice tiny little activities that a cell's going to do. But again, if you ever do that, if you ever use your ATP, you're left with the spent version, ADP. But we can use the energy from food to put it right back together again. And that is the ATP cycle. All right, there is a part 3 video. Even though there is cellular respiration in the title, that is not the focus of the video. We will be talking about fermentation, which is an alternative way to make ATP if you're a prokaryotic cell like a bacterium or a single-celled fungus like a yeast and you don't have oxygen around. So how can you make ATP when there's no oxygen, no ETC? That's what we're going to be talking about. Hopefully you'll watch that video. Thanks for watching this one.